Okay. Yeah, it's now recording now.
Okay.
Yeah.
Okay. Okay, so hi everyone. Thanks for coming to our Phosphatase Zoom group. I'm glad so many people are not zoomed out and are able to make it. So far we have it set up where it seems like people have kind of naturally signed up for once a month to present. So it seems like it's going to be once a month meeting, unless we get a bunch of new people that want to present. We do need somebody for July 10th, if anyone's interested. And let's see, so the way we have it set up are two 20 to 25 minute presentations. Hopefully we'll have lots of time for questions. If you have like a burning question during the talk, I think Caroline says you could put it in the chat. Oh, I guess I should say, I'm Jessie. Caroline is here. Can you wave, Caroline? Yeah, Caroline's there. She's the one that's mostly organizing things. But you can email us both if you have questions. So we are going to record some of these if the presenter says it's okay, and we'll post it to YouTube so people that are on a different time zone or have meetings and can't watch could watch later. We'll send an email around to the group and maybe also put it on Twitter. And also, I guess, just like spread the word. If any of you guys know other people that are interested in like phosphatases, the more the merrier, I guess. Is there anything I forgot, Caroline?
I don't think so. Yeah, just we'll do questions at the end, and then if you have questions during, you can put them in the chat, and then I'll like read through them at the end.
Okay.
And then, and if you have a question at the end, just make sure you unmute yourself.
Yeah. Did you mute everyone?
I think everyone already muted themselves besides me and you. Just
okay.
Two.
All right. So I guess do you want me to introduce Min?
Oh, wait, hold on. Yeah. Brittany has a question.
Yeah.
I just wanted to say, can you hear me?
Yeah, we can hear you.
I just wanted to say thank you for the Blackburn Lab for putting this together. This is absolutely amazing, and my group is super excited about it. So I appreciate all the efforts that you guys did. So thank you for that.
Yay. Okay, good. I'm glad like people are excited. And I guess also if you guys have any other like comments or suggestions on how to run this, either tell us now or you could email Caroline and I too. We're just kind of winging it. So,
yep.
Okay, so for the first talk, we have Min Wei, who just graduated from my lab, or just defended her PhD thesis. Thanks to coronavirus, she's still like in the lab, so we're keeping her for a while. And also Min is very nice and let me sign her up with just a week's notice. So she's going to talk today about the role of PRL3 and its substrates in T-cell acute lymphoblastic leukemia. Okay, Min.
Okay. Thank you, Dr. Blackburn, for the nice introduction. And today, I'm very excited to share my research story about a protein tyrosine phosphatase called PIL3 in T-cell acute lymphoblastic leukemia. So first, I'd like to start with a little bit background about this T-cell acute lymphoblastic leukemia. Acute lymphoblastic leukemia, also known as ALL, is the most prevalent pediatric cancer. There are two types of ALL, TLL and BLL, arising from two types of lymphocytes, T cells and B cells, respectively. Although T cell or TLL represents only 15% of pediatric and 25% of adult all cases, TLL is a more aggressive subtype compared to BLL. The prognosis for TLL patients is not bad, with 5-year survival rate higher than 80%. However, there's still several challenges for TLL in clinic. The first challenge is the central nervous system and bone marrow relapse. The relapsing patients had a very worse outcome, less than 30% in children and less than 10% in adults. Relapsing patients can survive. And another challenge is that current intensive chemotherapy regimens has long-term side effects. There is no target therapy available for TLL, which lags behind the development of BLL and other types of leukemia. Consider that most of our patients are children. So for the children at 5-year-old, this could mean more than 70 years or 70 years life with long-term side effects. So definitely we need more targeted therapies, which requires identification and characterization of new drivers for TLL. And our interest to explore this protein tyrosine phosphatase, PIL3, as a new therapeutic target in TLL, I trace back to my PI, Dr. Blackburn's large-scale screening using Zebrafish TLL model several years ago, where she found that PIL3 is significantly upregulated in the TLL clones with higher relapse potential, suggesting PIL3 might be involved in the cell proliferation or self-renewal capability of TLL cells. So what exactly is PIL3? PIL3 represents phosphatase of regenerating liver 3. There are 3 members in this PIL family, PIL1, 2, and 3. As the name suggests, they're also known as protein tyrosine phosphatase. As the name suggests, they belong to the protein tyrosine phosphatase, a big family. So they share the conserved protein tyrosine phosphatase structure as shown here. This is a P loop, and this is WPF DD loop. They are very critical for its phosphatase activity, and this is the polybasic region which can help target this PIL family protein to the cell membrane. And this proliferation motif is unique in PIL and not found in other protein tyrosine phosphatases. And this cysteine residue is very important for its phosphatase activity. So in my later study, I will use this phosphostate dead mutant, C104S mutant. PIL3 has been reported as a biomarker of tumor progression and metastasis in lots of different types of solid cancers. Here is the case study of PIL3 in cancer, came out in 2001, where people found the PIL3 mRNA expression level is significantly high regulated in the liver metastasis in colon cancer patient samples. And also later, people found that PIL3 correlates with poor prognosis with a bunch of different cancers. Here is an example in acute myeloid leukemia. The solid line is high PIL3 expression patients, less survival, and the dashed line is the low PIL3 expression patients, better survival. And also, in order to understand the role of PIL3 in cancers, a bunch of different cancer cell models and also the normal cells, HEK cells, are used to genetically manipulate PIL3 and to see how that affects cell behavior. You can see that when people overexpress PIL3 in those cell lines, you will observe increased migration, increased invasion, proliferation, and also metastasis in mouse models. On the contrary, if you knock down PIL3 in those cell lines, you will observe the opposite phenotypes. But most of these studies I'm showing you here are focusing on solid tumors. There are very few studies about PIL3 in blood cancers, and no one has reported the role of PIL3 in T-cell acute lymphoblastic leukemia, TLL. So I started to look at how PIL3 contribute to TLL progression 4 years ago, and recently wrap up the first part of my study and get it published in Oncogenesis this year. And here I would like to touch the major findings in my project. In my project, I used 2 different animal models to show that PIL3 can actually promote TLL migration and progression. The first model I used is the Zebrafish TLL model, where I overexpress PIL3 in TLL Zebrafish model and found that TLL onset is accelerated, and also the circulation of leukemia cells in the blood vessels in the Zebrafish are also facilitated. And in the second model, the xenotransplantation mouse model, I found that when I knock down PIL3 in TLL cells, those cells lose their capability to engraft and circulate in the bloodstream. And in the cellular study, I found that the SAC pathway is activated when you overexpress PIL3. And when you knock down PIL3, the SAC pathway is inhibited. The mechanism is via indirect de-phosphorylation in SAC. And also I found PIL3 is targetable in TIL when I use the JMS053, the small molecule PIL inhibitor to treat the cells. The SAC pathway is inhibited. And also the cell migration capability of those cells are also inhibited. And next, I would like to talk a little bit more about the interesting data in the Zebrafish TIL model, because I'm thinking most of the audience today are not familiar with this animal model. So here is the TIL Zebrafish model established in our lab, which is induced by oncogene MYC, which is a driving oncogene in human ALL. And the REC2 is a T-cell, B-cell specific promoter. M-cherry is a fluorescent protein. So when you inject a REC2 M-cherry plasmid into the zebrafish embryo at the single cell stage, you will see this transgenic fish will develop normal T lymphocytes labeled with M-cherry. This is the thymers. But when you inject REC2 CBIG plus REC2 M-cherry, you will see this transgenic fish will develop a full leukemia. The leukemia cells can expand out of thymers into the local tissues and enter the circulation. And this model can recapturate human TIL very well. So in my study, with the help of another PhD student in the lab, we did this experiment. In the control group, we injected a REC2 MYC plus REC2 M-cherry plasmid into the zebrafish embryo. And you will see an example figure here. Day 34 after injection, most of the leukemia cells are still confined within the thymers. But in the experimental group, because we also included REC2 PIL3 plasmid, so you will see this transgenic fish develop a more aggressive leukemia. A lot of leukemia cells already migrate out of thymers and into the local tissue and also enter the circulation. And this trend is also reflected by this survival curve. And the red line is the PIL3 overexpressing fish. The black line is the control fish. We can see that at day 60, all of the PIL3 overexpressing fish died, but in the control group, there's still 40% of them survive. Another very interesting phenotype we observed here is the circulation capability of the leukemia cells are different in two groups. Because the leukemia cells here is labeled with M-cherry, so we can monitor its circulation in the blood vessels in the tail fin. And this red dots representing circulating leukemia cells in the blood vessels. I want to show you a video here. You will see the circulating leukemia cells in the blood vessels. Yeah, those are the circulating leukemia cells in the blood vessels. So this is a video taken from the PIL3 overexpressing fish. And here's a quantification of the percentage of the fish we can capture the circulating leukemia cells in the blood vessels. You can see in the PIL3 overexpressing group, most of the fish, actually 5 out of 6 of the fish, we can capture the circulating leukemia cells in the blood vessels. However, in the control group, only 40% of them, we can capture the circulating leukemia cells. The rest of the fish, we can never find the circulating leukemia cells in the blood vessels, even in a very late stage of leukemia. So here clearly the phenotype that PIL3 overexpressing or knockdown in animal models is obvious to us. But since SARC is not a direct substrate of PIL3, so there's still a very critical question remain to be answered. What is the PIL3 substrate in TIL? So in order to answer this question, I studied the second part of my project. to identify PIL3 substrate. It seems that PIL3 substrate study lags behind its functional study. There are only 2 publications showing very solid evidence about the PIL3 substrate. The first one is done in colorectal cancer cell model, where carnitine 8 is found to be physically associated with PIL3, validated by Coll-IP and co-localization experiments. And 2 serine sites were dephosphorylated by PIL3, and leading to increased migration, invasiveness, and metastasis. A second study was done in colorectal cancer cell and HEK cells, where integrin beta-1 is physically associated with PIL3 and validated by a Coll-IP experiment also. The overall phosphorylation of integrin beta-1 is decreased by PIL3 overexpression, but the exact dephosphorylation site in integrin beta-1 was not reported in this study. And when you overexpress PIL3 in these cancer cells and HEK cells, the ERK pathway is activated, which leads to increased migration, invasiveness, and then metastasis. So the lack of PIL3 study in the solid tumors, I think probably due to the technical challenges about phosphatase substrate study. As we know that the interaction between phosphatases and its substrates is very transient. The phosphatase come in, remove the phosphate of modification, and then leave. So in order to capture the transient interaction between a phosphatase and its substrates, people normally use the substrate trapping mutant. And in the case of PIL3, we can use the C104S mutant to trap its substrate in the catalytic pocket. But here, I still have a concern about this substrate trapping mutant of PIL3, because according to the crystal structure study of PIL3, the catalytic pocket of PIL3 is very shallow. So I was wondering whether the binding between PIL3 substrate trapping mutant and its potential substrate are strong enough for the pull down experiment. So because of this concern, I turned to another approach to study PIL3 interacting proteins. This approach is called BiolD proximal labeling approach. BiolD2 is a biotinylate, which is an enzyme which can add biotin modification to proteins. So if you fuse BiolD2 to PIL3 and overexpress this fusion protein as a bait in TLL cells and culture the cells in biotin-free media, biotin-rich media, and this BiolD2 ligase will add biotin modification, as shown in the red dots here, to the PIL3 proximal proteins, including the directly interacting proteins or the proteins which do not directly interacting with PIL3, but just around PIL3. And then you can lysis the cells and get total protein and use strip adenine magnetic beads to pull down Biotinylated protein and then send these proteins to do mass spectrometry analysis. And MS will help you determine what those Biotinylated proteins are. So for my experiment, I generated 4 cell lines. I overexpressed BiolD2 PI3 fusion protein linked by HA tag, and I overexpressed them in the Jurkat HSB2. Both of them are TLL cell lines. And after puromycin selection, I will have JBP and HBP cells that can stably express this bait. And for the control cell line are overexpressed by BiolD2, also have a HA tag. And after puromycin selection, I will also have 2 cell lines termed as JB, HB for the control. So here is the Western blot validation showing that those bait protein are actually expressed in these cell lines. And I also did a validation of Biotinylated protein. Those cell lines were cultured in the biotin-rich media for 16 hours. Then strep-avidin HRP antibody was used to probe the biotinylated protein. We can see here a lot of biotinylated proteins are actually expressed after you culture the cells in the biotin-rich media. So after this validation, I did a large scale pull down and sent those biotinylated protein to a MS Coll in Stanford University. They help us to determine more than 3,000 biotinylated proteins. So here's the most challenging part of this project, how to figure out the real PIL3 interacting proteins from such a big pool of data. So the first thing we did is to use the CREPOM score to filter out the potential background contaminants. CREPOM is a public database where people deposit affinity purification MS data. So you can compare the protein you pull down with the proteins deposited in this CREPOM database and to get a score. For example, if you get a protein has a score equal to 0.5, that means 50% of the experiment deposited in this database also show this protein. So it is more likely to be a background contaminant. So in my filtering, I only show the CREPOM score less than 0.5. And most of them are in green color, which means CREPOM score is equal to 0. That means those proteins are unique in my product experiment. They are not present in this CREPOM database. So this first level of filtering brings down the number of this protein from 3,000 to 1,000. And the second filter is plot the fold change of biotinylation of these proteins in JBP or HBP versus JB or HB. The rationale behind this is, I believe, if a protein is a real PIL3 interacting protein, so it should have a higher level of biotinylation compared to the random biotinylation in the control cells. So here the two 10-fold line. This 10-fold line is in HBP versus HB, and this 10-fold biotinylation line is in JBP versus JB. So I would like you to focus this cluster, the up right cluster. These proteins are the proteins which showed more than 10 times biotinylation both in HBP and the JBP compared to their control cells. In this cluster, we found some interesting hit. The highest biotinylation level is the PIL3, which makes sense because PIL3 is fused to BiolD2, so it should have the highest level of biotinylation. And we also found two known PIL3 interacting proteins. One is CNMM3 and another one is CNM4. Also have, both of them have very high level of biotinylation. So those hits suggested that this cluster is a representative of PIL3 real interacting proteins. So how many proteins are here? There's still 288 proteins. So I put this 288 proteins to do the PANTHER analysis, which showed that 8 pathways are enriched. One of them is T cell activation pathway. Another one is PDGF signaling pathway. PDGF signaling pathway is also reported in another study which overexpressed PIL3. But now the 288 interacting PIL3 interacting proteins, potential interacting proteins, are still beyond my validation capability. So in order to further narrow down the PIL3 real interacting proteins, I went back to the substrate trapping mutant. As I mentioned previously, this substrate trapping mutant might not be a very ideal approach to study PIL3 substrate. But I was thinking maybe it can still pull down some strong interacting proteins. So I overexpressed this substrate trapping mutant in Jurkat cells and did a pull down using flat tech magnetic beads, and sent those pull down proteins to MS Coll and to see what kind of interacting proteins can be pulled down. And surprisingly, these pull downs still identify more than 1,000 proteins. But when I compare this 1,000 pull with the 288 common hits, I only found 43 overlapping. And among the 43 overlapping proteins, PIL3 and CNNM4 is still there. And there are several hits which are very interesting to us. One is LCK. LCK is also a SAC family kinase and involved in TCR activation, T cell activation. And another protein is LAT, which is also a component in the T cell activation pathway. And the RIGTOR is a component of mTOR pathway. So next, I began to validate those findings using Western blot. Here, I did another strep evidence pull down experiment, pull down biotinylated proteins in the JBP cells versus JB. But after pull down, instead of MS, I use a Western blot to probe the interesting hits here. The first one I probed is LAT. We can see that although LAT expression is high in the input, but after strep evidence pull down, it was not present. So that means the LT biotinylation is not real. It is a false positive hits identified by BiolD by ID2 approach. And the second is a RIGTOR. Although RIGTOR expression level is very low in the input, but after strip evidence pull down, it was enriched in the JBP, but not in the JB. And LCK, we can see the enrichment of LCK in the JBP is significantly higher compared to that in the JB. So we identified two real biotinylation protein. One is RIGTOR and one is LCK. And this biotinylation validation only tells you they're around PIL3. They're not necessarily directly interacting with PIL3. So next, I did another pull down experiment. I also used this PIL3 substrate trapping mutant and used magnetic beads to pull down PIL3 and its binding its potential substrate, and use the Western blot to probe it. And we can see here, the LCK is pulled down in both the substrate trapping mutant and also pulled down by the wild type PIL3. But RIGTOR was not pulled down, suggesting that RIGTOR is not a direct interacting protein of PIL3, but LCK might be a direct interacting protein with PIL3. And I also found that the LCK phosphorylation level is regulated by PIL3 overexpressing. So here we found when you overexpress PIL3, the wild type PIL3 in the Jurkat cells, the phosphorylation of tyrosine 505 in LCK decreased, but recovered when you overexpressed the mutant one. And another phosphorylation site is tyrosine 394. Its phosphorylation is also decreased in PIL3 wild type overexpressing Jurkat cells, but also recovered when you overexpress the mutant PIL3. So this evidence suggested that the LCK is very likely to be a PIL3 substrate. So what is LCK? What is the function of LCK in the T cells? So LCK, as I mentioned, is also a member of SAC family. It is an essential component in the TCR signaling. TCR signaling is involved in TCR activation. So when this is the TCR receptor complex, when it binds to a peptide presented by antigen-presenting cells, it will recruit LCK to the TCR receptor. And then LCK will phosphorylate TCR receptor and also another kinase called ZEP70. And ZEP70 will further phosphorylate other proteins involved in the TCR signaling pathway and leading to full activation of T cells. But how LCK contribute to TLL progression has not been reported. So I would like to stop here and summarize my studies about PIL3. In this study, I found that the BiolD-based proximal labeling is a feasible approach to study PIL3 interactomes, and LCK might be a PIL3 direct substrate in TLL. And for my future direction, I need to further determine whether LCK is a real PIL3 substrate or not. For this purpose, I will do a reciprocal IP overexpress LCK in the TLL cells and put on LCK to see whether PIL3 is also associated with LCK or not. And also we'll look at if I knock down PIL3 in TLL cells, how that changed the phosphorylation of LCK, especially at those two phosphorylation sites. And another very important question is we would like to know how PIL3 LCK interaction affects TLL progression. So lastly, I would like to thank my lab, my mentor support. We have a lot of fun together. This is a picture we took for celebrating 2090 Christmas. Thank you. And next, I can take questions.
Okay, yay, nice job. So I guess if anyone has questions for Min, you can unmute yourself and ask, or type it in the chat and Caroline and I can read it if you want to do it that way. Anybody?
Yeah, I've got a question.
Yeah.
So in the kind of first study that you presented with the MIC story, it almost looked like the phenotype had maybe two different phases to it. It's like you kind of see that in your survival curve, and you kind of see that in the circulation as well, that like early time points kind of match, and then as you go further along in time, that's kind of when they separate. So I don't know if you have any thoughts as to whether this is kind of two different phases, kind of like an acute versus a long term, or you know, any of that, and how MIC, like, do you think the LCK and MIC story are connected in this context?
That's a good question, but we haven't explored it yet. But we do think PIL3 might interact with MIC. We have another project in the lab which studies how PIL3, collaborate with MIC to drive TLL progression. We have some preliminary evidence about.
And would that be driven through direct MIC phosphorylation changes?
Probably not by phosphorylation. Okay. So Jesse's hypothesis is might be PIL3 might interact with at transcription level.
Yeah, we're finding like here. So someone asked if PRL3 is localized, where it's localized. And so we're finding it. So it has that prenylation motif. So it's at the membrane, but we also see it in the nucleus when we have GFP fused PRL3. And so part of what Min's doing is what's it doing at the membrane, maybe with LCK, because LCK is at the membrane. And then another, well, if we can find someone to do it in the lab, I guess, would be like, what's PRL3 doing down in the nucleus? But yeah, I guess we do notice that the fish, there could be different effects in the leukemia depending on, where PRL3 is and what the timing of it all is. So I don't know, the more we get into it, the more we're like, what is even going on? It gets more and more complicated. So.
Welcome to the MIC field.
Yeah.
Or the PRL field.
Yeah, right.
Yeah, so I was wondering, So first of all, I think also it's a great talk, great story. I was wondering about the LCK story. This is very interesting. Have you also thought about using phosphoproteomics to compare your biotin ID data on a more global level, maybe through using an inhibitor to have some acute changes that you could measure? So just to get a broader picture.
We have done used the proteomics trying to look at the global phosphorylation change in the cell lines. We are still in the middle of analyzing those data. It's more complicated than I expected. It's not only the phosphorylation level change, but when I overexpress like PIL3 in the TL cells, the phosphorylation site also change. Do you know what I mean? Like in the control cells, maybe tyrosine 5 is phosphorylated, but when I overexpress PIL3, it's another tyrosine site was changed. So it's a very big data. So we're still in the middle of how to make those phosphorylation change make sense to us.
Interesting. Thank you.
You're welcome.
Okay, anybody else? Okay, going. Okay, so thanks again, Min, and thanks for everyone for coming to the meeting. We have a sign up, I guess, in 2 weeks if anyone wants to sign up for then, or otherwise the next person that has signed up to present is the first week of June. So we'll, Brittany. So we'll see you guys then. So, all right.
Thank you. Bye-bye.
See you, everybody.
It was good to see everybody's faces. Bye.
Yeah.
Bye.
Okay, how do I leave?
Caroline, do you want me to send you the after?
Send the recording